Hello everyone, this is our second installment of doing our video lecture with our anatomy and physiology class. And we are technically 5 weeks into our class, which puts us at our cells chapter. So the first night that we met, I handed out a series of packets to you. This should be the final packet that you have not done anything with up to this point. So there, you should have a packet that is just simply titled cells, and that's the one that we're going to be working through this evening. So if you don't have that packet with you, put this on pause, go find that packet, and then that is the one that you will want to have with you and take notes on. Please make sure that when we have our next face-to-face meeting, you remember that you are taking your quiz over the anatomical terms, 2 parts. In one part, you will be given the word and you will have to match it with the definition. And in the other part, you will have a picture and you will have to apply the word. There will be a word bank and it will not be an identical picture to the ones that you were given in class. The terms obviously don't change, but it's going to be look slightly different, like I said, in the picture form from what you guys had. So just so you're aware of what that quiz is going to look like when we come back on the 28th. All right, so moving forward with the cells. When we talk about cells, this is really the first time where we get to the basics of biology. So anatomy and physiology is a branch under the biology umbrella. And when we talk about cells, cells are the most basic unit we have in biology. So every branch of science has its smallest form. In chemistry, it would be the atom. Biology, it's the cell. So this is really the first place where you can have life existing in a confined little unit. And when we talk about the cell, that's what we are talking about. You have something that is functional enough that it can sustain everything that is necessary for life, all in the confines of this one little cell. Now, that is for your most basic organism. You guys know very well that when we talk about something like the human, we are talking about trillions upon trillions of cells that it takes to keep a human fully functioning and doing everything that it needs to do on a daily basis to survive. So the number of cells that you have within an organism is going to vary greatly, but you can have everything that you need that is necessary for life in a single-celled organism. So when we talk about cells and we apply that to the human body, if you put this into perspective, it's really kind of amazing because at conception, fertilization, you have a single sperm from the male body, you have a single egg from the female, and those two are going to unite. And from that, you are going to get a series of cell divisions that are going to continually take place until you get to the human. And like I said early on in your formation, you're talking about those two cells, those two become four, four become eight, eight becomes 16, and that process continues until you are at the form that you are currently at. And like I said, that number is in the trillions. I hesitate to put a number to that because there is no definite number. If I asked you guys to look that up on the internet and come to class with a number of cells in the human body, every single one of you would probably have a different answer. So depending on the resource that you look at, whether that's a textbook or an internet resource, that is going to be very different because how people count things as it relates to the number of cells in the human body does differ greatly. So just know that we are in the trillions. Now, if I say that number, the trillions, you guys know that that's a huge number. But if you put that into perspective, if you were to simply count to a billion, so we're not even at the trillion level, if you were just simply to count to a billion and you counted at the rate of counting a single number every single second until you reached 1 billion, it would take you over 33 years to do that. So that's a huge number in itself. And then we're actually going above that when we talk about the trillions. So the organization that goes into the human body to keep you guys working and doing everything that you need to do is really very impressive. That you have the brain in your body that is able to direct all of that, monitor all of those cells, make sure that they're doing what they're supposed to, and when they're not, figure out how they're going to redirect those cells ultimately to get them back on track and make sure that you survive. So when we talk about that process of those two cells combining together and starting the process of developing a human itself, one thing that you have to understand is in those early phases, the cells are all alike. There is no differentiation between the cells. It's simply going from 2 to 4, 4 to 8 on up until you are continuing that process. Now, at some point, the cells begin to differentiate. So a group of those cells are going to now start transitioning to become your heart. Another group of cells is going to start transitioning to become your stomach and your muscles and your intestines. And the thing that baffles scientists right now is they have absolutely no idea what causes that to happen. Now, the potential with that information, if they could figure out what in the cell actually triggers them to go from being just these blank slates to getting the information that, okay, now you guys are going to become the heart, you're going to become the brain, you're going to become the spinal cord. If they could figure out what causes that, then ultimately they would be able to harvest cells early on in development, and they would be able to program them with what they wanted those cells to become. Think about the potential with that. If you could harvest cells, essentially stem cells, don't have any programming yet, take them out of a developing body, and then program them for what you want them to become, then a lot of the illnesses that we have, a lot of the diseases that we currently are battling are no longer existent because they could take cells, develop them into what they need them to be, and transplant them into those people that are struggling with neurological disorders. Things like Alzheimer's, dementia would be a thing of the past. And so being able to eventually come up with that answer as to what tells the cells, okay, you're going to go from being, like I said, a blank slate, and now you're going to start forming a particular body structure. The possibilities with that are absolutely endless. Now, the reason that stem cell research is such a hot topic issue is that in order to get those cells that are still in that blank slate sort of state, typically you have to get them from a developing embryo. And if you're taking them from the developing embryo, you are potentially ending, or you are ending that embryo's ability to then continue on with life. So there's a lot of people that have some very strong opinions on that, as they should. Now, you guys sitting in the classroom right now or sitting wherever you are, you have stem cells within your body. So why don't we then go and take them from adults? If we have stem cells as full-grown adults, why not use that as our source? Well, the problem with stem cells in adults is that our stem cells are embedded in existing tissues. They're not in these large clusters or large groups like you have them in a developing embryo. And so they're very easy to get to in a developing embryo, very, very difficult to harness and get from an adult because you would literally have to go in and pick them out one at a time. So not very efficient in that process. Now, when we talk about cells, a couple of things that I want to identify right off the bat is that a lot of times people have this misconception about what a cell actually looks like and how it actually functions. And so if I asked you to imagine a cell, and we talked about this in class last time we met, I said, what shape is a cell? And everybody goes to that circle shape, as you should, because 99% of the time, that's what we're going for. But what you need to understand about a cell is that, number one, cells are compartmentalized. Now, what does that mean? That means if you think about your home, most homes are not just open areas inside. You have walls that are going to create different sections or different parts to your home. So you have walls that create a bedroom, a kitchen, a bathroom, closets. And the reason for that is there are very specific things that need to happen in each one of those rooms. And so you create those walls to make sure that whatever activities need to be done in that room are separated from other areas of the house. Cells have to work the same way. And the reason for that is your cells are tiny little factories. And this is kind of a misconception that people have. They think of cells as basically being building blocks, that we are using them as these brick-like structures to build bone, to build muscle, to build the human body as a whole. And while that's part of what they do, your cells are tiny little factories. They do work 24 hours a day, 7 days a week. And ultimately, what your cells are trying to do is they are trying to produce all of the things that you need in order to stay alive. So when you think about that muscle tissue, the proteins for that are actually made in your cells. The hormones that you guys need in order to regulate certain things within the body are made within your cells. And then as they are produced, the cells ship them out to where they actually need to be used within the body. So while cells do form your bones and form your other tissues and your organs, ultimately what they do once they're in place is they continue to produce the products that you guys need in order to stay alive. So if we need to kind of make that shift in how we view cells, they are not just these open bubbles where all these parts are just kind of floating around and going wherever they want. They are very structured, like I said, very much like your home, and certain things take place within certain areas of the cell. And the biggest reason within the cell that we need to keep that compartmentalized is because a lot of what happens in your cell is a chemical reaction. And so if you have these chemical reactions that are taking place, we talked about those positive and negative charges and how when you get those positive and negatives to interact, you ultimately get the reaction to then take place that you're looking for. Well, if everything was allowed to just float wherever it wanted to within the cell, what would happen is you would have the wrong positive and negatives interacting, and you would get chemical reactions, but not the ones that you want. So by keeping everybody compartmentalized in their own parts of the cell, you can regulate when and where chemical reactions happen and ensure that the right chemical reactions are happening and that something's not getting used up in the cell because it was able to interact with something it shouldn't have been able to. So cells are compartmentalized, very structured in their internal form, and then they're also, start thinking of them as being factories, that they are producing products nonstop to make sure that your body has everything that it needs in order to fully function. So in talking about the cell itself, we're going to start talking about the cell membrane. And the cell membrane is really a very dynamic structure. It is going to encompass the entire cell. So it encircles the entire cell, but the outer membrane itself is made up of fats. So you've probably heard of a lot of different names for the cell membrane. It's called a lipid bilayer, a fluid mosaic model. You got the cell membrane, plasma membrane. You have all these different names talking about the structure that is going to surround the cell as a whole. Excuse me, what you need to understand is that it is made up of all these tiny little lipids. Now, a lipid, if you draw a circle somewhere on your paper and then draw two tails kind of coming off the bottom of that circle, that's what a lipid looks like. A nice round head, two tails extending off of it. Now, what happens is the head of one lipid is going to line up next to the head of another one, and so on and so forth until they make a full circle around the entire cell. Now, inside of that, there is going to be just the opposite. So you're going to have another layer of lipids, but now the tails from another lipid are going to meet up. So you have the head and the tails coming down, and then inside of that, you have the head from another lipid and the tails so that they are facing each other. So the tails face in, the heads face to the outside. I will put a picture of this on the board, so if that's not cementing right now and you're not getting a clear picture of that, don't worry, we'll come back to it. But you have this double layer of lipids that are forming a barrier around the cell. And essentially what they're doing is they're holding the contents inside the cell in, and they are creating a barrier to keep those things outside of the cell outside. Now, things can pass through those lipids because even though they are creating this barrier, they're not physically attached to each other. So there is nothing connecting one lipid to the next. If I asked you guys to stand up in the room and stand shoulder to shoulder with each other, that's essentially how the lipids are. They touch each other, they're right next to one another, There is no physical bond that actually links or ties them to one another. So if you have a molecule on the outside of the cell and it needs to come in, if it's small enough, it will simply move between those lipids and make its way right into the cell itself. Now, the reason that the cell membrane can stay together, even though it's not physically attached, is that the head of the lipids, or those fats, are what we call hydrophilic. Now, hydro, always and forever, is going to indicate water or liquid. Philic, or phil, P-H-I-L, that is always going to mean to love. So when you see the term hydrophilic, you break it down, it means to love water or to love liquid. Hydrophobic, hydro still means water. Phobic comes from phobia, which is to fear. So this is water hating, or it fears water. The head of the lipid loves to be exposed to water, and the tails hate it. So they always stay positioned so that the heads are going to stay to the outside, and the tails will always stay to the middle so that they are never exposed to any type of liquid. And that's what holds this all together. Now, the other thing is, when I talk about the fact that your cell membrane is made up of lipids, sometimes people just throw that out there and it's just kind of taken at face value. Nothing in the human body happens by chance. So if we're talking about the cell membrane being made out of fats, there is a very real reason for it. And you guys were all exposed to this if you ever saw The Wizard of Oz. Wizard of Oz, they throw water on the witch, and what does she do? She dissolves into a puddle on the floor. If your cell membranes were made out of anything other than fat, then that would be the potential. Because so many things dissolve in water that if your cell membrane was made out of something that dissolved in water, every time you guys came in contact with liquid, you would dissolve very much like she did. So taking a shower would not be a possibility. Jumping in a pool would not be a possibility. Getting caught out in the rain would be a very different experience. So the structure of that cell membrane being made of fat is very, very important to our survival. So that cell membrane, kind of talking about that point as to why that is so important. If you go on to the second slide that's called cell membrane, when we talk about cell membranes, the other thing to keep in mind is that it's in the human body, it is what we call selectively permeable, meaning it picks and chooses what it wants to get in and out. So if it's a molecule that is floating around the outside of the cell and the cell needs it in order to build something, it can pull that into the inside of the cell so that it can use it. If it's not something that it needs, it will leave it to the outside. And so selective, it's picking and choosing. Permeable, being able to permeate or pass through that cell membrane, it picks and chooses what it wants to come in and out of the cell. Now, the one thing that the cell has to be careful of is, remember, those lipids are not linked together. So it has to be small molecules that it's allowing for it to move in and out, because if it's too large and it tries to push through those lipids, it'll basically rip a hole in the cell membrane and it will break down and the cell will be destroyed. So in the case of larger molecules that need to pass through, what the cell actually does is it creates these protein channels. So every so often in this lipid bilayer, you are going to have protein channels. Now, again, that's kind of hard to envision when we're talking about something that's microscopic, but if you think about it in the terms of our classroom, when they built the classroom, they have the 4 walls making up the classroom. And if you were the right type of molecule, even though it looks like those walls are solid, if you were small enough, there's holes in those walls, you would be able to pass through the wall. Now, you are not able to do that because you are a larger molecule. And so what did they do in order to allow you to be able to come into the room? They put doors in there. There's a door at the front, there's a door at the back, and you guys are able to easily pass through those doorways to get in and out of the room because you're too big to pass through the walls. So essentially, those protein channels are just doorways that were built into the cells to allow those larger molecules a proper structure to be able to come in and out without destroying the cell as a whole. The other unique thing that the cell does is it uses a series of carbohydrate flags in order to be able to recognize. So on the surface of the cell, there are these little thread-like extensions that stick off of the surface, and they are going to do a couple of different things. Primarily, they're there for cell-to-cell recognition. So if you think about the way the body is put together, a lot of your cells are going to be touching other cells. Your heart and your lungs come in contact with each other. Your stomach and your intestinal tract come in contact with each other. Your blood is circulating through your body. It comes in contact with tons of structures. So if your cells have these little threads kind of sticking off the surface, these carbohydrate flags, as your cells bump into each other and the carbohydrate flags from one cell touch the carbohydrate flags from another cell, because they were both made from your body, you're able to recognize those carbohydrate flags. And the cell knows, okay, this is something we made. It's supposed to be here. It's okay. In the case of something invading your body, so you get a bacteria or you step on something with bare feet and you get a shard of glass in your foot. Now, as your body is coming, or you get a splinter, as your body is coming in contact with those foreign carbohydrate flags, so let's say you've got that wood splinter or you've got that bacteria floating in your body, they're going to have their own carbohydrate flags, but they look very different. And so when your cells come in contact with their carbohydrate flags, your body doesn't recognize it. And now it knows this is something that doesn't belong here, and we need to make sure that we take care of it before it has the chance to cause any problems in the body. So cell-to-cell recognition, making sure that what's in your body was made by you and is supposed to be there. The other big thing that it does is it makes sure that your body knows how much growth it has or it can allow. Because your internal organs do come in contact with each other, when you grow and give them more space, they need to grow as well. But there has to be some sort of regulation to make sure that they only take up the room that is allotted for each one of your internal organs. So what happens is you grow and develop, and now they have more space. They will start to grow and develop. But if you have your heart and your lungs, and as they are growing, now those cells are going to come back in contact with each other. So when the carbohydrate flags from your heart bump up against the carbohydrate flags from your lungs, now they know, okay, we've taken up the room we have. We need to stop producing cells for continued growth. And so they will shut down the new cell formation. They will quit growing until you have another growth spurt, and then they know they need to grow again. And that process just allows them to really be able to monitor how much space they have for that growth. So cell-to-cell recognition, and then being able to monitor that growth and development to make sure that that is regulated appropriately. So the next thing that we have to talk about are the organelles. And I would just like to start out by saying these are the major organelles. These are not the only organelles that we have within the human body. And so if you've taken other classes, you might have heard of some others. If you take other future classes where you specifically are looking at cell biology, you'll probably hear about some that we didn't talk about. These are the ones that we need to talk about for the purpose of our classes. So starting out with the organelles, if you just look at that word, organelles tells you little organs. And if you think about the structure of the human body, we have certain organs inside that perform very specific jobs to keep us alive. Your heart's going to pump the blood, your stomach is going to process the food, and your lungs are going to move oxygen in and out. And everything that those internal organs do to keep you alive, you essentially have to have the same sort or the same concept happening within the cells, but it's on a much smaller level because of their size. So small organs that are going to perform pretty much the same jobs and tasks of your internal organs to keep your cells alive and doing what they need to do. So the first one that you have on the list is cytoplasm. And cytoplasm is it's slightly thicker than water. It's a clear fluid, and it fills the cell, and it's going to do a couple of different things. Number one, it helps the cell maintain its shape. So it fills all the extra area within the cell to make sure that it gets that nice round structure that we think about. And then there are some things that move in and out of the cell. And that cytoplasm is going to help make sure that they're able to move in and out and reduces the friction. So it just helps with that process of moving material through the cell. Now, when we think about cytoplasm, if you guys look at the room that you're sitting in right now, you have probably some chairs, some furniture, other things, maybe a TV, computer. So if you think about all the space that those things are taking up, that's essentially the equivalent of the organelles. So you have these organelles within the cell, they take up a certain amount of space, but they don't take up all the space. Now, what happens is if you are still considering that room that you are in now, any space in that room that is not taken up by furniture, other materials, is going to be filled with air. And that's the equivalent of what happens within the cell. Any space that is not taken up by an organelle, we're going to put that cytoplasm in there. So if you are kind of on that visual side of things, that's the best comparison that I can give you for cytoplasm within the cell. So the next organelle that we have to consider is chromatin. Chromatin is going to be a form of DNA, and DNA will actually change shape depending on what's going on with the cell. So we have chromatin, we have chromosomes, and we have chromatids. So chromatids ends TID, slightly different than chromatin. It's all your genetic information. The different terms that we have, chromatin, chromosomes, chromatid, is going to simply indicate the shape that that genetic information is in. So when it's in chromatin, when it's in that form, it is very relaxed, it's very spread out, it almost has a speckled-like appearance to it, but essentially it is your genetic information, all the information for how to make you. But the cell is not actively dividing, and so it just kind of relaxes and spreads out and doesn't take on a particular shape or form. The next one that we have on the list are the ribosomes. Ribosomes are hugely important in the process of what the cells do because ribosomes are going to be key in producing proteins. Now, what happens is proteins are going to basically form all structures in the body. When we start looking at DNA, there's a protein base. You have protein channels in your cell membranes. You have protein in your muscles. You have protein in your internal organs, in your blood. There is protein in everything that makes you. And so these ribosomes are essentially creating proteins so that you can build muscle, so that you can build DNA and your cell membranes and all the other structures that need protein. The key is they're building different types of proteins because the protein that you need in DNA is different than the protein you would need for muscles. So ribosomes are going to vary in number inside the cell because if you are dealing with a muscle cell, it needs to have more protein than other types of cells in the body. So muscle cells have a huge amount of ribosomes. Some of your other cells would have very few. So depending on how much protein they need, the number of ribosomes in that particular cell type will vary. Now, ribosomes, if you were to look at them on a picture, they're going to look like small round circles. And they don't really have any key marks or identifying features. You just learn to kind of find them and recognize them. The two major places that you would find ribosomes, number one, you can have free ribosomes, and they just float around in the cytoplasm. They are able to go where they want to go within the cell, and that's kind of one of the exceptions to, we talked about everything kind of having a fixed location. Of course, there's always an exception to the rule, and your free ribosomes are that exception. The other type of ribosome are going to be attached to a organelle that we're going to talk about here in just a minute, which is the endoplasmic reticulum. Now, if ribosomes are attached to the endoplasmic reticulum, they are stuck to the outside, and then it becomes rough endoplasmic reticulum. It's abbreviated RER, so rough endoplasmic reticulum, and it just indicates that that is a section of endoplasmic reticulum that is going to contain those ribosomes. And they, like I said, they just stick to the outside. If you look at a picture, you'll notice it looks like the endoplasmic reticulum has little freckles attached to the outside because you are seeing those ribosomes that are stuck to it. We'll come back to that in just a second. Underneath ribosomes, the next one that you guys have on the list talks about the nucleolus. Now, I'm going to kind of pull two together here. You have the nucleus, which is typically the largest organelle that you are going to have in any cell. So the nucleus typically is going to be at the center of a cell. And the reason for that is it's a protective issue. The nucleus is the brain of the cell, regulates everything that's going on in the cell, monitors the condition of the cell, makes changes when needed. So what your brain does for your body, the nucleus does for the cell. But your body has the skeleton, which is able to offer protection to your brain and your other highly vital organs. And the cell has a little bit of that, but not enough. So in order to offer an added layer of protection to the nucleus, we're going to put that nucleus right at the center of the cell and help protect it as much as possible. Now, within the nucleus is where you would find the nucleolus. And typically when you see a picture of it, you'll see the nucleus. And then at the core of the nucleus, you're going to have a small, dense region, darker in color. That's the nucleolus. The nucleolus is going to have a variety of different jobs. One of the jobs that it's going to carry out is it's going to be another place where protein is made. And when you think about it, if you have all these structures within the human body that are dependent on protein, it would make sense that you would have more than one place that is going to produce protein. The other thing that the nucleolus is going to help with is the production of RNA. RNA is a genetic material that we have that is able to move and go to more places than what your DNA can. Again, DNA is very, very important. It's the blueprint for how to make you. And so it doesn't get to leave the nucleus, but that RNA is kind of a smaller version of DNA and has a little bit more mobility within the body. Underneath that, you have the endoplasmic reticulum. A lot of times it will be abbreviated as ER. If you can abbreviate it as ER and that's going to make sense for you 2 weeks down the road, a month down the road, great, use ER. If you're going to look at that 3 weeks from now and you're not going to remember what that means, go ahead and write it out. I know it's a little bit longer. But the endoplasmic reticulum is a very unique structure. It attaches to the nucleus. So we just got done talking about the nucleus being right at the center of the cell. And the endoplasmic reticulum is going to form an attachment to the nucleus in multiple places. And basically what it does is it creates a network that is going to take those products that were made within the nucleus. So those proteins, the RNA, anything else that might be formed within the nucleus, and it is going to start directing it where it needs to go in the cell. So it talks about a network of vesicles that are made up of membranous sacs. That's kind of hard to visualize. So again, if you are kind of visual in your learning, think about when you go to St. Luke's for class and you come in the front doors and you start to navigate a series of places. You have to go in the elevator or you might go up the stairwell, and then you're going to navigate through the hallways that are eventually going to lead you to the door of the classroom where we meet. That's what the endoplasmic reticulum does. It is nothing more than a series of hallways in the cell that helps take those products and make sure that they get delivered where they need to be. So you have your path that you follow from the front door through the hallways to get to the classroom. And the endoplasmic reticulum is going to do that same thing for any of those products that were created. And then they just channel them and deliver them where they need to be. Sometimes that's going to be somewhere in the cell, and sometimes they will go ahead and they will network them all the way out to the cell membrane and actually deliver them outside of the cell. So it just depends on where they're going to be used in the body as to where they get delivered. Now, two types of endoplasmic reticulum. If it has ribosomes, it's rough ER. If it does not have ribosomes attached to it, it's called smooth endoplasmic reticulum. And you can identify that in a picture. If it's a clear surface, nothing attached to it, it's your smooth. You can see those ribosomes dotted on the outside of it, then that is your rough. The way that you can tell the endoplasmic reticulum apart from some other structures that look similar is because it is going to form an attachment directly to the nucleus. Then we have the nuclear envelope. The nuclear envelope is going to be a protective layer that you have around the nucleus. And so very much like the cell membrane that we have, you have this layer around the nucleus that is going to contain all the contents of the nucleus. And the biggest thing that it's trying to hold within that nucleus and keep in place is your DNA. So if you look at the description, it talks about the fact that you have thousands of pores on the surface of this nuclear envelope that wraps around the nucleus, but those pores are maintained and kept very small, and your DNA is actually very large in comparison. So that DNA is held captive in the nucleus, but the other products, the proteins, the RNA, the other materials that are being made are small enough that they can move through those pores, leave the nucleus, make their way into the endoplasmic reticulum, and then start getting delivered to where they need to be. And the last one that we have on that list are the vacuoles. Now, vacuoles are going to be basically storage tanks for liquid. And on here it says water, but it can be different types of liquid as well. With the vacuoles, it's hard to distinguish. They are larger than ribosomes, but they're also circular in nature. So when you're looking at a picture, you're trying to label it, and basically you're going to have these small round circles, and then you're going to have these larger circles. And trying to tell which one is which, you just basically have to make a decision on size. If it's very small, it's a ribosome. If it's a little bit larger circle, it's probably a vacuole. And that's just something that over time, as you get more practice, that will become a little bit easier, but they're both circles shaped. So just distinguishing size-wise if it is ribosome or if it's a vacuole. Now, the reason that we want to contain that water is because of those chemical reactions. So early on, I mentioned the fact that you have all these chemical reactions that need to take place and happen within the cell. And you want to make sure that any extra liquid that's in the cell is going to be contained, because you don't want to dilute the interior of the cell, because it can change the ability of those chemical reactions to be able to take place. So if you think about if you've ever made a batch of Kool-Aid, so you make up the Kool-Aid and you make it according to the directions, it should taste the way that you want it to. But if you take those directions and you double the amount of water that you put in that Kool-Aid and now you taste it, it's going to have a very different taste to it, and you're probably not going to care for it as much. So likewise, if you have all that extra water that is allowed to just flood through the cell and go wherever it wants to, it's going to dilute those contents and things are not going to work the way that they should. So vacuoles are very important. It sounds like they're kind of boring. They just hold this excess water or liquid, but they're actually very, very important, because if the cell would get too diluted, unfortunately, things are not going to work the way that they should. So I'm going to stop this first video here, and I'm going to pick up with the second video so that through the week, as you guys are watching these, you can kind of break it up a little bit. That is perfectly fine, but I am going to end this first one. Please make sure that you look for that second one that will be posted as well.